Dear friends, good morning. Today we can discuss about antigenic determinant. Antigenic determinant means epitope, haptens and adjuvants. First we will see about antigenic determinant. The site of antigen which reacts with specific antibody is known as antigenic determinant site or epitope. Epitope is a small region which exhibits a very high degree of stereospecificity and also chemical specificity. The number of epitopes differ from antigen to antigen. The site of an epitope is 25 to 35 angstrom unit and the molecular weight ranges between 400 to 1000 Dalton. Each antigen contains more than one epitope. Generally, epitopes are found on surface of antigen, but on denaturation and unfolding have revealed some hidden epitopes in addition to functional surface epitope. The number of antigenic determinant per molecule of antigen is called as valency. The picture shows antigen, the surface of antigen which have the epitope. The epitope antigen which binds to paratope of antibody. Antigen binding site of antibody are called as paratope. Antibody binding site of antigen is called as epitope. Epitope generally present in antigen, paratope generally present in antibody. The epitope paratope are complementary to each other. Binding sites are complementary to each other. Next one, types of epitope. There are two types of epitope. Those epitopes which interact with T cells are known as T cell epitope. And another one is B cells means B cell epitopes. Next move on to B cell epitopes. The size of this epitope ranges from 6 to 7 amino acids and it should not exceed this. Generally, B cell epitopes having the 6 to 7 amino acids alone. B cell epitopes should be hydrophilic in nature. Hydrophilic means water soluble one. Only then it can reach the blood. Epitope may be sequential or non-sequential segment of antigen. Sequential or non-sequential means the arrangements are either sequential or non-sequential one. It should be located in the flexible region of antigen. The B cell epitopes located flexible part of antigen. Next move on to T cell epitopes. The size of T cell epitope is 7 to 20 amino acids. It should have two regions. One is to react with T cell alone. Next one is reacts with MHC aggregate. The region which reacts with the T cell are hydrophilic in nature, whereas aggregate is hydrophobic in nature. Next move on to haptens. Haptens, Karl Landsteiner used various haptens for his study. Haptens may be defined as molecules that are not immunogenic but that react with performed antibodies of right specificities when combined with a carrier molecule. Chemical coupling of hapten to a larger protein or called as carrier yields an immunogen called as hapten carrier conjugate. The carrier molecule are called as hapten carrier conjugate. Generally, haptens are incomplete antigen. The hapten which combined with carrier molecule then only become a immunogenic property. Biologically important molecules such as drugs, hormones can act as a hapten. Next one, uses of hapten. Antibodies can be raised for immunological assay of hormones and also drugs. Already I told hormones, drugs are example for hapten. Hapten molecule alone not produce the immunogenic response, which conjugate with carrier molecule then only become a active one. The hapten carrier conjugate, this complex only induce the immune response. The picture shows the individual hapten does not produce the immune response. The hapten molecule combined with the carrier molecule, then hapten carrier conjugates the conjugated form of hapten, then only it produces the immune response. Next, move on to adjuvants. Adjuvants means hardware to help or agents to potentiate the immune response, either humoral or cell mediated immunity. The adjuvant molecule generally induces the humoral and also cell mediated immunity. Adjuvants are administered along with antigen. Generally antigen injected means along with adjuvant also injected. Adjuvants are often used to boost the immune response. Generally the adjuvants which induces the immunity or immune response. Example, BSC. BSC means bovine serum albumin injected in mice, which induces the immune response, normal one. BSA combined with adjuvant then injected to mice, which increases the immune response 5 times. 5 times means 5 folds increase the immune response. As like one more example, BSA injected to rabbits, which produces the low immune response. The BSA conjugated with adjuvant then injected to rabbits, which heightened the immune response. Heightened the immune response means which produces the more amount of response. So the adjuvant we are using the boosting the immune response. Next one, effect of adjuvant. Effect of adjuvant, otherwise called as functions of adjuvants. First one, prolonged antigen persistence. Prolonged antigen persistence means the antigenic property prolonged with it will be located. Example, aluminium potassium sulfate. Aluminium potassium sulfate, otherwise called as alum. Alum and also antigen injection results in alum precipitates slower release in injected site. Effective time of exposure of antigen few days to several weeks. Generally antigen injected means the immune responses are quickly disposed. We are combined with alum means the antigenic property, antigenic response is more time exposure there. Next one is Fred's adjuvant. Fred's adjuvant, two types. One is complete fruit adjuvant, another is incomplete fruit adjuvant. First we will see about incomplete adjuvant. Antigen in aqueous solution, mineral oil and emulsifying agent. Here emulsifying agent means manide monovolate we are using it. These three combinations in antigen dispersed in oil into small droplets surrounding the antigen. We are injected these combinations means in this way it will be dispersed in the surrounding the antigen in injected site. The antigen release is very slowly from the site of injection. Next one, complete antigen is highly effective adjuvant containing heat-killed mycobacterium and additionally ingredients like incomplete adjuvant. Here complete adjuvant means we are mixing the heat-killed mycobacterium as well as aqueous solution and mineral oil and emulsifying agent. These are we are using in incomplete adjuvant. These are the things combined with the heat-killed mycobacterium we are using in complete adjuvants. Here muramyl dipeptide cell wall component of mycobacterium which activates the dendritic cells, macrophages, thereby making the fruits complete adjuvants for more potent than incomplete adjuvant. Here we are comparing incomplete adjuvant and also complete adjuvant. Compared to incomplete adjuvant, complete adjuvants have the more potent one. Second one, enhanced co-stimulatory response, which enhances the stimulatory response. For example, T helper cells recognizes the antigen associated with the MGUS2 molecule on the membrane of APC. APC means antigen presenting cells. Here antigen presenting cells means dendritic cells, macrophages and also B cells. These three cells are antigen presenting cells. Third one, induce granuloma formation. Stimulate a local chronic inflammatory response. Granular formation which stimulate the inflammatory response. Fourth one, stimulate lymphocyte proliferation non-specifically. Dear friends, thank you for your support.